Looking at our friendly model here, what am I going to call this covering? Sarcomere. That is the cell membrane. And what is this kind of lacy stuff on the sides of it? Yes, the sarcoplasmic reticulum, which is like ER in other cells. What are these yellow things? T-tubules or transverse tubules. Going to be very important today. On each side of it, some storage sacs called cisternae, which are part of the Golgi in other cells. All right, then we take this off, and we're now in a functioning unit called a sarcomere. Okay, and so these ends are the, okay, and so these blue things are the, and what are they? They're filaments. They're filaments, which is the protein or macromolecule level. Okay, these are the actin, which are also myofilaments. And so now, right by the Z line, where I only have actin, is which band? I band. Then where I have an overlapping from here is the A band. Actin drops off, I have pure myosin, H zone, and then we're attached in the center to the, but myosin is attached over to the Z line by titin or connectin, where actin is attached where? At the Z line, but not over here because it's going to have to move. Okay? All right, so today it's about getting into the physiology of this. We're going to compare ourselves starting out this morning with a steam engine. Any of you ever been on one? I don't even, anybody been on Spirit or Jasper? Have you been to French Lick, done the Polar Express or any of those?
Yeah, train ride.
Yeah, I don't know whether they're still steam operated or not, but geez, way back in the 70s, I think it was, the place to be, place to go shopping, ladies, was in New Albany. There was a store there called the Fashion Shop. We didn't have any close by, and they would take a train from Princeton to New Albany on Sunday afternoons, and they let you shop at the fashion shop. There was ice cream parlors and stuff downtown, and then you could come back. And so my girlfriend and I decided that sounded like a good day. You know, it's a July afternoon, not really thinking. Okay? Number one, no air conditioning on the train. Number two, steam train. Okay? Note to self, steam produces heat. Okay? And so when we look at the efficiency of a steam engine, it says that it can convert 10% of its energy to go down the track, and the rest of it is lost as heat. So that says it was pretty warm. How do we compare an efficiency to a steam train? Well, a little bit better than that, because 25% of our energy is used in muscle contraction. But remember, anytime we use glucose for energy, what's going to be a byproduct? Heat as well. Okay? And so 25% of the chemical energy of glucose is used for muscle contraction, and the rest is converted to heat to maintain a normal body temperature. And of course, we appreciate that a lot in the wintertime. Maybe we would like to reduce that a little bit in the summer. Ironically, muscle is 80% water. Now, most of you have been probably in a period of time in your life when you've lost some weight, and we always say it's water weight. Okay? I hope I'm not losing muscle. Well, in reality, that's where a lot of the water is coming from, is out of that muscle. But the rest of it are those two important proteins that we call actin and myosin. There's also a small amount of fat. Is there any place we have that we can't have that? Some glycogen, which is what? What's glycogen? Stored sugar. Okay? Glucose becomes glycogen before it becomes fat. There were two places that we stored glycogen. Remember where they were? Liver and muscles. So glycogen will be present in our muscles. And two phosphorus-containing substances. ATP, which stands for what? Adenosine triphosphate, and phosphocreatine. and sometimes referred to as creatine phosphate. And so let's take a look at that reaction for cellular respiration again. Obviously, it's always important. C6H12O6 plus oxygen is going to form carbon dioxide, water, ATP, and heat. And of course we said that we can take in sugar and store it first as glycogen. And remember that occurs in our muscles and liver. And then of course we can still convert it to fat. So we want to take a look at what's happening in a muscle contraction. Glycogen, oxygen, phosphocreatine, ATP are going to decrease. If you haven't eaten sugar, you're going to break down the glycogen that's stored. You could possibly break down the sugar. Essentially what we're saying is things on this side of the equation are going to go down, things on the other side of the equation are going to go up. Okay? And so these things are going to decrease, but even to be able to go through this reaction cost us some energy. So we're also going to spend some ATP and phosphocreatine. Now, what's going to get increased? Well, obviously doing work creates carbon dioxide. Okay? Now remember, we have a choice here. This breaks it down into two C3 molecules. Do you remember the name of them? Yes, pyruvic acid or pyruvate, whichever you want to call it. And that was what kind of process? Anaerobic. Which occurred where? Yes. Do you remember the name of that reaction? Yes. And how much ATP did I get? Two. And so this was my decision mark, because if oxygen was available, I got to go where? Krebs and electron transport chain. But if oxygen was not available, what did I do? Fermentation, which created lactic acid. So as I break this down, not only do I increase my carbon dioxide level, but as my oxygen gets depleted, I'm going to increase lactic acid production as well, which is so important because that causes us to stop, right? How many of you have ever had lactic acid buildup that you're aware of? Okay? And usually runners, will experience that. Last night when we were playing ball, all of a sudden Allie started grabbing his legs, and he's like, "Grammy, I got a cramp." And he said, "Are cramps real?" I'm like, "Are you feeling it?" Okay? And he said, "What is that?" And I'm like, "Well, let's have a talk, honey." And he said, "What?" And I said, "Well, there's two reasons that cause cramps to happen in your legs. Number one is you've played so much, you've used all of your oxygen, and your body's telling you need to rest because that lactic acid builds up. But another reason you have cramps is because you're not very good about eating fruit. And a lack of potassium can also cause muscle cramps. The little one, that's almost 3, he'll eat 3 bananas in one day. Can't get the big one to eat 1 banana in a week, much less long. And so he's very, very picky about how he eats, but he's also a little bit worried about what he eats being healthy. Somebody has been driving this in his head. So he'll say, "Grammy, look at my plate. Is it healthy?" And I'm like, most 6-year-old kids don't really care, whether it's healthy or not. But that kid sits and eats salad like it's his best friend. And I'm like, okay, I'm glad you do that, but it's not the normal reaction that I see. But I can't hardly get him to eat oranges, bananas, any of that stuff that's going to provide that potassium. So lactic acid, it is very real as an increase as we do exercise. But then, what's going to happen? Okay, as ATP is produced over here, remember we don't want to keep it at this if you're not going to do any work. So what do? We break it down into subparticles, ADP, which is what? Adenosine diphosphate, and a phosphate group. And so remember, once we need to make ATP, all we have to do is hook these back up together and you get ATP. So you've got this cycle of ATP to ADP, ATP, ADP. And so whether you're using it or not, you haven't lost it. You're keeping that intact. So essentially, we're depleting the things on the left and increasing the things on the right in this reaction. All right, so let's start by trying to get working through a physiology of a muscle contraction. And so first off, it says there were 4 characteristics that a muscle possessed. Do you remember what they were? Excited. You have to get excited first. That says I do what? To a stimulus, okay? So I can respond. And then I got 1 of 2 things that can happen. Contractibility or extensibility. And then we hope that we can, elasticity will take us back to the normal position. So for a muscle to be able to contract, the first thing it has to experience is that excitability. It has to be able to respond to a stimulus. So now, what's the definition of a stimulus? Any force that affects your excitability. How do you wake up in the morning? Is an alarm going bzz, 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 bzz? Is it a cat licking you? Right. Is it mom turning the light on? Hey, all of us have different stimuli that we respond to, and some of my kids, I swore I was going to have to get the alarm clock that brings the hammer out that goes kapoom, kapoom, you know, because no matter, I mean, they could lay there and hear an alarm, and I used to worry when they go to college that they would never get to class on time because they could never hear that alarm to get them up. So we all, maybe, are any of you very light sleepers? Okay? And that kind of happens more so even after you have children. You're like, okay, you just don't, you know, sleep as sound, but it could be something as tiny as a feather or a fly or whatever. That could be the stimulus that causes a muscle to contract. So for a muscle, the stimulus will be an electric impulse. So now let's take a look. Hold on. I mentioned this. Am I doing anything?
Okay.
I mentioned this last week that a nerve does not go and pierce a muscle. It lays across the top of a muscle like this. And so remember, there is a gap between the nerve and the muscle, and if there's a gap in your electric current, what's going to happen? It won't go. Okay? If I cut a wire and there's a gap between the 2 wires, we're not going to have any movement going on. And so I've got to be able to go across the gap. So that gap is called the neuromuscular junction. What are we looking at? The hole between a nerve and a muscle. So, and of course you recognize this is a sarcomere, and this is the sarcolemma here. Now this is the end of my nerve. And so my nerve has all of these branches here. They are called axons. We'll talk about those a little bit later in the nervous system. But we're going to look at one of these blown up here. And of course you can see mitochondria there. Okay, you can see this is the muscle side. The yellow represents the nerve side. And that blue in between is the hole between them. That is called the neuromuscular junction, where the nerve and the muscle come together and form that gap is referred to as that neuromuscular junction. All right, now what we want to focus on is the muscle side. Now notice how the sarcolemma starts to fold. And I remember when I was a kid, I've never been a decent artist, but you know, here was my head, my eyes, you know, my nose, but this would be as good as hair got, okay, for me. And so that's what the folded sarcolemma looks like to me. Like you have just done that weaving. And so instead of being a nice flat membrane, that membrane has folded and created all of these little dips. That part of the membrane then is referred to as the motor end plate. And so a specialized area on the muscle fiber made it a folded sarcolemma, and of course you can see there's a lot of mitochondria down here as well. And so now that is the motor end plate. All right, now how are we going to get across this gap? Electricity is coming down my neuron, or my nerve fiber, and we get to this gap, and I'm supposed to stimulate a muscle, but my electricity won't go across the gap. So what happens is the electricity triggers these little vesicles. Remember, vesicles are usually storage sacs, and they have a neurotransmitter in them. Neurotransmitter says he's going to keep the message from the nerve going across the gap. And so stored in here are these neurotransmitters. So the electricity causes these to be released, and now these dark blue particles represent the neurotransmitter, and it goes across the gap and tells the muscle what to do. So you could have a muscle not firing because the nerve doesn't work. You could have a muscle not firing because maybe there was no neurotransmitters to come across, or we'll see what happens on the muscle side of it. And so anytime somebody doesn't have muscle movement, you can't just immediately say, it's my muscle. There's a lot more involved to it. So the first thing we have to really see is we received a stimulus, and it has traveled down this motor neuron, so it triggered the release of the neurotransmitter from these vesicles, and it now jumps that gap and starts a new impulse on the muscle side. Okay, so far so good? Makes sense? All right, we're talking about skeletal muscle today. So the neurotransmitter in the muscle that I can actually control is called acetylcholine. Acetylcholine, neurotransmitter in skeletal muscle stored in the synaptic vesicles. The synapse is this gap, and that's what we call it. So the vesicles on the neuron side are called synaptic vesicles in the end. It increases when the nerve impulses reaches the junction to transmit the impulse across the synapse. All right. So now, synapse or synaptic cleft is that gap between the axon, which is the end of the neuron, and the folded sarcolemma. What happens when this neurotransmitter goes across here? What does he do? He acts on the muscle, and ACH is the abbreviation for acetylcholine now. And so that impulse acts on the membrane of the muscle as acetylcholine combines with acetylcholine receptors on the motor end plate, causing a new impulse to be generated. This impulse travels both directions down the muscle fiber through the sarcoplasmic reticulum and will diffuse into the sarcoplasm of the muscle fiber. All right, a few things we need to be aware of. First off, acetylcholine is the neurotransmitter. And it is like anything else that's hormonal, and we'll talk about this in A&P too, but you can have 100 different hormones flowing in your bloodstream. But your ovaries, ladies, won't respond to all 100 of them. They'll only respond to 4 of them. Unless you have a receptor on a tissue, doesn't matter what that hormone is, it won't respond. And remember, receptors are typically fibrous proteins found in that phospholipid bilayer that we learned about earlier. And so for me to be able to have a muscle contraction, I have to have a receptor for acetylcholine, so when it comes across the synapse, it bonds and now makes a new impulse. So are there diseases out there where acetylcholine receptors are destroyed? Absolutely. Okay, as we start looking at muscle atrophies, and it can be one of the issues that we see. And have you heard of the disease myasthenia gravis? Never heard of that one? And it has to do with acetylcholine receptors as well. All right, so we received a stimulus. It's traveled down the motor neuron. It has triggered the release of acetylcholine, because we're talking about skeletal muscle here, from the synaptic vesicles. It's jumped the synapse, bonded to a receptor on the muscle side, generated a new impulse going how? Both directions through the sarcoplasmic reticulum. So we've gone across the gap and we're now going through the SR to the muscle in both directions. All right, now let's take a look at another definition. Let's see if I can find this. A motor unit, okay? Here is a neuron, and notice how a neuron will have several branches. Now each one of these is a muscle fiber. Now what's a fiber again? A cell. And so it's going to take a lot of cells to make a tissue, and a lot of tissues to make a muscle. So if you want, like some of your muscles may have as many as 400 fibers. If you want every cell to work together, you better have a branch touching a cell so that you have maximum impact. So you don't have one neuron per cell, you have one neuron that triggers several cells within that. Now I kind of think this looks like dynamite getting ready to go off whenever you see this. And so a motor unit is the neuron plus the muscle fibers that it's controlling. So you'll see in a muscle contraction there will be several motor neurons trying to get this muscle to do its work. All right, next definition here. The ratchet or the sliding filament theory. Contraction is brought about by the sliding of the thin filaments at each end of the sarcomere toward each other between the stationary thick filaments. Thick filaments are attached to the M line. They're not moving. Contraction is brought about by the thin ones coming closer to the center, okay, in the stationary thick filaments. And so we call that the ratchet or sliding filament. When the thin filaments move towards each other, it draws the Z lines closer together, and the sarcomere has now shortened. When contraction is great, there is a dense zone in the center of the A band as a result of the overlapping of the thin filaments. You know, normally there's an H zone in the center of this that does not have actin overlapping. But when contraction is happening, now my A band and H zone really become the same thing because we've got a total overlap of the actin and myosin filaments at the same time. And so the movement of the thin filaments occurs because of connections with the cross bridges of the thick myosin filaments. Well, let's take a look at this a little bit closer. Here is my myosin, and here are the heads on my myosin. You see those little golf club-like things. And then you will see, let's see if I can show you, my thin filament, the actin, they look like olives. And can you see that my olives are kind of light purple, but the pit of them where the pimento would be is kind of a fuchsia color or whatever. And so what's going to happen is one of these heads is going to stick in one of those holes. And do you know how a cog on like a bicycle chain works? You know, you've got this projections going around, and then you've got these links on your chain. And so what happens is that gets in a link and it keeps pulling it. Well, it's the same thing that's happening here. One of these heads is going to get in one of the holes, and eventually he's going to ratchet it to the center. And so that's where it gets its name, the ratchet or sliding filament theory, from these heads connecting in the actin holes. And so the myosin cross bridges move like teeth on a cog of a watch wheel on the surface of the thin filament, creating what we call the ratchet theory or the sliding filament theory. Okay. All right, so let's get this much down pat so far. We start with the what? Why am I going to do anything? I've got a stimulus. And it travels down what?
Neuron.
And at the end of the neuron, it triggers the release of what? Acetylcholine, which is going to diffuse across the synapse, bond to a receptor on the muscle side, generate an impulse where? both directions through the sarcoplasmic reticulum. What's going to happen next? It's going to cause this series of reactions where the myosin heads are going to get in the actin holes, and we're going to bring the sarcomere closer together. Okay. Now, are you starting to see why I drive anatomy so hard? Because physiology is a no-brainer when you know what it looks like. I ain't trying to say all of this without taking the time to explain this. Probably not going to make too much sense. But where is it at for you? Well, Dinah, I know you work ortho. I don't know what you're going to do whenever you get finished. But most people don't ever see the internal body. Most people are going to be dealing with blood tests that are going to tell them these things aren't happening. Acetylcholine's not present. Potassium's out of whack. Calcium's out of whack. And we've got some enzymes called CPK, CK, creatine kinase, troponin, all kinds of things that tell us what's happening in a muscle, and now you should have a picture that says this is what's going on. Because those blood tests will tell you how this isn't moving. All right, so that's our first little bit of our physiology that we're working with. All right, so now we want to look at the chemical composition. Remember, let's see, do I have, I don't think I have a picture. No, I'm going to go back to Tuesday just a minute. Spent a lot of time on this. This was what level? Organ.
Organ.
This was
Tissue.
this was
Cell.
and these are organelles, and these are macromolecules. Okay, the covering around a whole entire muscle is a fascia. Okay, then the covering around a muscle underneath that is called a epimysium. A covering around a tissue is paramysium. What's a tissue level called? Fascicle is the word for tissue. Okay. Yes, it is a tissue layer, but it's a fascicle. And so now the covering around the cell level is called endomysium. And then we equated these things to being like a where's my cell? There we go. Okay. We equated these guys to being PVC pipes.
Yeah.
Okay. And so, like, this is one PVC pipe. Okay. And so this represents what level? These are fibrils. Each one of these is a myofibril. These are myofilaments. Okay. And so what we want to look at now is what is the chemistry inside of one of those PVC pipes, the myofibril group. Okay, now let's go back to today. All right, first off, last week, for quick purposes, we said these red ones were called what?
Active.
And the blue ones were called?
Myosin.
To be politically correct, we should call them thick and thin. Because that's not all they are. It's just actin and myosin. So today we need to get more familiar with that. So let's start with the thick filament first. Well, you can pretty well guess the most abundant protein will be myosin. But if we look at this on the model, it is composed of two twisted protein strands. Okay? And so we have a rod-like one that is helical. But then we also have these two globular heads, or golf clubs. And so they're called the cross bridges. And one of their little heads is going to hook into the actin hole, and the other one's going to grab some energy. Okay, so one binds to actin, the other one binds to ATP. So when I say, explain the